Hey Eric, this is your soil sample. We're having a look. We're zoomed out to 40x, the least magnification we'll use under the microscope. We see this. This is the top of the food chain as far as the soil. It's not a nematode, but it's a white worm, I believe. It's a good thing, not a bad thing. Another nutrient cycler. And right around him, we're going to see some nice branching fungi up here we saw. I would believe that's part of a root there we're looking at. We'll see things like this. That's humic acid. We'll see fulvic acid as well. That's these colors. As we zoom in, we'll zoom in and see bacteria and fungi. Here's more branched fungi. A lot of these are minerals. That's that sand we're seeing in your soil. So a lot of this is minerals, which is good for aeration and will break down over time and feed the soil as we get more organic matter in it. This is 400x, the most I'll magnify, but we'll see bacteria at this level and other protozoa. Let me change our lighting a little bit. And the bacteria are these dots and rods we're seeing. I got to zoom in and out through this layer. And we're seeing a nice amount of different colors, shapes. These are rod bacteria, and these are dots. The dots are cocci. These are bacilli. They're all good. What we don't want to see is a spiral one. That would be anaerobic. So everything so far just looks great. We're not seeing huge numbers, but we're seeing good soil here. What I might do is pause so this doesn't get real. Oh, there's a nice piece of branched fungi there to look at. Here we can see this is decomposing fungi, saprophytic fungi. You see the different sections, the septa in it. Again, the bacteria all around it. That's a spore from one, I believe, there. Again, good, real good signs. We want to see this. You don't see this usually in someone's backyard in their lawn. They just see mostly bacteria. It's a nice piece of branched fungi there. Saprophytic fungi. And a good amount of bacteria as well. But I'm going to pause you here and I'll have a look around and we'll just see who we can point out in here. We didn't have to go far. I shouldn't even stop the camera. Right next to it was this protozoa. Two of them. So this is a flagellate moving around. And this is a shelled amoeba. So they're both at second level in the food layer. We start off with the 5th trophic level, which is the fourth, would be microscopic animals. So we have bacteria, fungi, are kind of the first and second layers, similar sizes. Protozoa, which there's going to be multiple types. There's going to be 3 types. This is a flagellate spinning around here. It's actually one member with a tail. There's stuff hooked to the tail now, but it'd be one circle with a tail we see, or different shapes with a tail, a single flagellum. That flagellum is what makes them a flagellate. That's how you distinguish it. This is a shelled amoeba. If we look, we can see kind of a shell around it. So it's a testate amoeba. The test is the shell, and that round part is the mouth. It is going to feed on the bacteria and or fungi, depending on what type it is, or other organisms that can fit in its mouth. So all that is cycling nutrients. Those are good things. So this is good to see these organisms in there. We haven't seen any that we don't want to see. No signs of anaerobic environment. Be back in a minute. So this is a nematode. There's 5 different types of nematode, but this is what we're looking for. We can tell by the mouth parts on it. This is a bacteria. Oh, geez, it's very touchy when I'm zoomed in. This is a bacterial feeding nematode. You can tell by the lips and mouth structure on it. It's either resting or dead. We can see the tail there. We can even see the bacteria inside its belly as we zoom in and out. We're zoomed way in right now. That's the top of the food chain again. We have bacteria, fungi, multiple types of protozoa, microarthropods, and then nematodes. And then arthropods would be things like spiders, things we can see with the naked eye. So these members of the soil food web, we have aerobic and anaerobic. Everything we've seen so far has guided us that you have a very aerobic, which is what we're looking for, full of oxygen, with lots of that soil life in there. We're seeing a decent amount of diversity. Let's look around and see if we can find some more of those testate amoeba and whoever else, the flagellates that we can see. So we saw the humic acid. This is fulvic acid. More yellow. The other one had more of a reddish to it. Another good sign, another good part of the soil food web. This will be consumed by those other organisms. And that nutrient will be shared amongst all those members of the food web and given to the plants. This we can see soil organic matter all being held together by that fungi. That's a part of its purpose in the soil, is to hold everything together so we don't have that erosion. We'll zoom in. And here we see the different fungus at different levels and layers in that organic matter. So this was something that was once alive, one of those trees, bark, plants living, being broken down by the fungus, the bacteria, the protozoa, mostly the fungus and the bacteria. But they're taking that nutrient created in photosynthesis and returning it to the soil to be refed on by the plants, animals, the plants. So the breaking down of that organic matter by the fungi that we see holding it together and making it stable, but also feeding on it. And also those bacteria. There's other fungi that actually will connect out in these areas as long as you don't till your soil and go back to those plant roots, carrying those nutrients through these fungal pathways. And that's why we don't till. Camera there. This is a saprophytic fungi, a decomposing fungi, but same concept. We can see, oh, and there's a flagellate moving around right there. Let's see if we can get into that. He's moving fast. And here's a shelled amoeba, like we were looking before. We'll zoom in on this a little bit. Again, that's that third trophic level, the bacteria, the fungi, and this is a protozoa. And in the protozoa, we have amoeba, shelled and unshelled. The unshelled are hard to see. Flagellates. Then we have ciliates. And ciliates, we are not seeing any, and that's good. We can see a few and it'd be all right. We'd show us we're on the edge. But a cilia is a sign of an anaerobic environment. So all this is great. We're seeing lots of bacteria, those different shapes and sizes that we want to see, and colors. The dots and the rods, no squiggly ones. We've seen lots of protozoa in here. Flagellates, hard to get a good shot of those, but the shelled amoebas we've seen. We saw that white worm and that nematode, so the upper trophic level. We haven't seen any rotifers or small animals, but I'm sure they're around. This is a single sample we're looking at. I'll look a little bit more here, and I'll add a couple pictures I've taken as well. There it is. Look through the eyepiece to be sure. See if we get better color on it. You can see the body and the tail at the back of it. And when it moves around, it uses that tail to propel itself. So that's second, third level again. Bacteria, fungi, protozoa. You saw something exit it. It could have been a bacteria actually going through it, around it. That's that nutrient cycling again. He's eating those bacteria and fungi around him, traveling around eating. I think what I'll do now, so if you've seen everything you need to see in here, I'll do a sample of the worm castings so you can see those as well that we're using. I've got one of those mixed up. You can really see moving around well now. It moving around well now. So I'll put a sample of those worm castings on and we'll be back. Okay, we're in the worm castings now. We got a real active flagellate. Having a hard time staying with him. Oh, we got a glimpse and that was all. There's a testate amoeba right behind him. There's a testate amoeba. Oh, there he goes. A chunk of nice chunk of fungi and just a real diverse bacterial content we can see in here. So in these worm castings, we're zoomed in all the way now, 400x. More different types of protozoa and bacteria and fungi. We just see a real diverse mix of everybody in there. And that's what we're doing with this. We're inoculating the soil so they can reproduce and populate in your soil. There goes another nice fungi or protozoa. There's a flagellate. There's another testate amoeba. Let's zoom out. We'll go to 100x. There's that flagellate moving around there. We'll follow him up, see what else we see. Nice colorful shelled amoeba. The flagellate right in the background. Testate amoeba we can have a look at. See that shell going in and out across it. The mouth on this end. Large number of different size bacteria around it. One of those yellow testate amoeba. A spore by it. There's some fulvic acid. More shelled amoeba. You can see the insides of this one real good. The mouth on that end. Again, a tremendous amount of, oh, there's a flagellate going through. You see the tail out the back. It moves so fast. There's a different layer in there, so they're hard to see. Hard to believe the different layers. We're looking at a tiny drop of water, a portion of that, but that's what it is. There's a good look at a Narcella amoeba, like I said. We're not seeing the mouth on that, but we can tell that's a shelled amoeba. So that's called Narcella. There's multiple types of those. There's the Narcella I was looking for. There we go. There's the Narcellas. Back to our picture. Very similar. Shelled amoeba of some sort. More nice branched fungi. The fungal spore up there. Some really nice rod bacteria and cocci bacteria. Cocci and bacilles. It's another testate amoeba. I can't tell if it's breaking down on one end or what's going on with it. A little different shape. More rod bacteria, a piece of fungal hyphae there broken up. Another large bacteria, and I can't think of the name of it. More fungal spores, lots of bacteria and fungi around us. We are seeing smaller flagellates in there. They're too small to zoom in on. There's a tremendous amount of life. So again, that's why we use these worm castings to inoculate with. We put this in the water, sprinkle it lightly, and inoculates that soil. See the diversity of shapes and sizes in there, all having different functions. These are all the decomposers cycling those nutrients because it came from compost, a long compost. It's a year-long process we have. Okay, I'm sure there's been plenty in there to see. There's a real nice diversity of these. You see fungal spores. Like I said, the fungal hyphae everywhere. Lots. We can't see the bacteria and protozoa from there, but when we zoom in, we see those everywhere. We're very proud of these castings. So we'll put that seed down. We'll put the castings over top. We'll inoculate the seeds with it first. We'll make a nice tea with it. We'll spray it all over everything. And we'll be off to the races with it. It'll do a wonderful, quick job of healing that soil. See you soon.